excuse me, I'm just gonna...
Okay. The plastic capped ones are glucose. Glucose. So I've always put the glucose first. The metal capped ones are lactose.
Yeah.
And so you have one sugar or the other in each tube, and you put different bacteria. So this one had E. coli, this one has Enterobacter, Proteus, alkaligenes. Okay. Now you're testing to see, can E. coli in this set of tubes, can E. coli digest glucose through a process called fermentation? If it can, then it's going to break the glucose down into acid.
Okay.
The acid is going to cause the phenol red to turn yellow because that's a pH indicator.
Yeah.
So it turns yellow, boom, it's positive.
So technically, this one's actually more simpler than that one.
It is.
So
Fermentation happened here. So I can just go through each tube and show. Okay, it's yellow and it's turbid and has gas. Fermentation definitely happened of glucose. E. coli fermented glucose. Here we have yellow and it's still turbid, meaning it grew. And there's acid. We don't know about gas. Yes,
because the
E. coli.
the lactose ones do not have,
The
they
durum.
don't have the gas thing.
Yeah, there's no Durham tube.
Okay.
Right, so no gas collection. You know, we could have put a gas tube in there, we just didn't. Okay, so positive for lactose fermentation.
Positive.
Enterobacter are both glucose and lactose fermentation occurred. Okay, Proteus, glucose fermentation occurred, but lactose fermentation did not, and it is a true negative because it grew. It's turbid. So remember, if there's no growth, you can't say it's a negative. It's inconclusive.
Okay.
Okay. So positive for glucose fermentation, negative for lactose fermentation. This organism, no yellow, but they definitely grew. You can see all the cloudiness. Okay. So negative for glucose fermentation, negative for lactose fermentation.
Okay.
Okay. So positive, 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 positive. This one just happens to have more gas than the other one. I mean, gas is just sort of, there's not much significance other than it helps us to characterize which organism is which. Some organisms characteristically produce gas. Some produce a lot of gas. Like, I mean, look how much gas is in there.
Yeah.
Okay. And some no, even if they're positive. So that's just kind of a side note. Okay. Positive, 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 negative, negative, negative. If it's yellow, it's positive. Pretty much just guaranteed for fermentation of whichever sugar is in that tube.
And so this one is negative.
No, that's positive. If
Okay.
it's yellow, it's
But
positive.
but the whole thing would be negative, right? Or
Yeah, like
just
would
this
we
one?
have to know which one, like if it was positive for glucose or positive for
Well,
lactose?
I would tell you. Okay, most likely the exam. I'll just test one sugar. The exam might look like. Actually, let's take a little more variety. Most likely the exam will look like this. So forget about these.
Okay.
Here would be, there would be like organism A, B, C, D. And we could even put like, we could have an organism A, B, C, D, E. This is an uninoculated control. So if it's yellow,
So these would be both positive.
Positive.
These would be negative, and this would be
Negatives.
no growth.
and that'd be no growth, which is called inconclusive, or no result.
Okay.
So no growth would be now you're recording the result, no growth, but then you have to interpret the result, which is inconclusive or no result.
Okay.
Okay. So positive, positive, negative, negative. no
And so
result.
for these, like if we were asked to explain, it'd be turbid and yellow.
Right. Turbid and yellow, turbid and pink,
Pink.
and then no
Clear.
growth, clear, no turbidity, no growth.
Okay.
And then this would be here just so you can compare, like just like an uninoculated tube, so that you can see how clear is and how turbid, just so you can compare turbidity to say clear.
Okay.
And then I would just say, oh, all of these tubes, all these broths contain lactose, or I could say, oh, glucose or whatever. The gas Durham tube thing, it's just there to see if gas was produced or not. It's not really a big deal. It's either there or it's not.
Okay.
Yeah.
Cool.
So this is obviously not inoculated.
It's neutral. So this
Oh,
is basic.
no.
Basic.
Okay, let's say that, what if I told you that this tube was inoculated and the bacteria were incubated for, say, 48 hours? Like you put bacteria in there, you came back 48 hours later, and the tube came out like that.
You wouldn't know.
What they call? I'm trying to figure out which one's basic, which one's neutral. Because I know they're really close.
This is neutral, okay?
Okay.
And these are alkaline, they're basic.
Okay.
And these are acidic, okay? But what I'm trying to say here is that obviously this uninoculated tube,
Right.
it looks just like this tube. But what if I said this tube actually received a microorganism? You know, someone took a loop and stirred some bacteria into there, incubated it for 48 hours, and came back and it looked the same as this. Like, how would you record that result?
And then we're inconclusive.
Yeah, you'd say inconclusive. Say no growth occurred, therefore it's inconclusive, or therefore we cannot record a result, a useful result. We say no result. Yeah, it would require a retest.
Because of that whole reversion thing, right?
No,
Or is
actually,
that design
because
nothing to
of the
do
no
with it?
growth thing. Okay.
Well, it's-
Reversion would be assuming that like, okay, so then with respect to reversion, the appropriate question would be, what if you were looking at these tubes
I don't want to cover
after
up the record.
incubating
I don't know if it's this
the tubes for a week? Would you be able to rely upon the results? No, because how do we know that these tubes that are pink right now
Don't turn me on.
weren't yellow, like, say, 4 days ago?
Yeah.
You know, that you're supposed to read the results at, like the book says, like 24 to 48 hours at the most. Yeah, a
So
week is like, oh, the sugar could have run out.
Reversion is when pH changes back and forth.
Right, so it starts out neutral, okay? Because we can say this is like day one. Starts out neutral at 7. As the bacteria begin to use the sugars, if there was a time-lapse photography, it would eventually turn to orange and then down to yellow. Okay? Now, that's because the sugars were used and acids were made and the pH went down. But if you left this tube in the incubator for like, instead of 2 days, what if you left it in for like a week? Then the time-lapse photography would show you, if you could have like a little camera in the incubator, it would show that it would turn from yellow to kind of orange to red to pink. And so the pH would go 7, down below 7, and then after a week, it would climb up to like 10 or 11, and you'd have a pink turbid tube like this. That wouldn't be a true negative, because at one point it was positive, and you just didn't read it at 24 to 48 hours.
Yeah.
So reversion is just a concept that has to be watched out for, because you know, if you're running a lab or you're working in a lab and you're doing these kinds of diagnostics, there's an importance in saying that I must take that reading during the window of time where the the colors can be trusted.
Did we get tested on reversion? Just by looking at it? Or knowing what sugar was used and what the time period is?
I haven't put a reversion test on the exam before. I put it in quiz questions, and I put it in.
And I mean, that would be pretty simple.
It's in my
too,
question
'cause
set.
The key would be,
Just think of time, the
Yeah.
timeline of over 24 hours, it could
Yeah.
revert to reversion.
It's a valid. I mean, it's.
Casein proteins
I think it's a
glucose,
fair
sugar.
question if I wanted to put it
Yeah, it's
on
a
a
good
test.
question, actually.
It's
Yeah, it's a
it
good
requires that you understand that pH
Timeline.
changes with over time based on the food that's first available, which would be the glucose, which is preferred, and then if that runs out, bacteria can actually shift their attention to this other food.
Casein protein.
Yeah, or if I was to ask,
Lactose.
you know, why did these tubes, even though they're negative for fermentation, why did the bacteria grow? Oh, well, because there's other food in there. There are proteins in there, you know, and it's just so that you understand that there are two food sources, and glucoses or sugars are always used first if the bacteria can use them. Some bacteria have the genetic ability to use them. Some of them, like alkaligenes, have lost the ability, and so their only choice is the protein. And whenever proteins are used, the pH goes up. Whenever sugars are used, the pH goes down. That rule will apply the rest of the semester. Like, that's just a basic understanding of cellular physiology. Is that sugars will acidify, will bring about acidic environments, whereas proteins will cause the environment to alkalinize. Yeah. So, yeah, and these basic experiments will help you to apply that understanding to like a lot of other things we're going to do. And they'll take less time. This took a lot of time to explain today, but
That's
they'll
all new.
start to get a little
A lot of
more
this,
efficient.
so yeah, it's a lot of information, so.
Yeah, but
I know, today was so.
Today was horrible, wasn't it?
Yeah. See, I think you guys are going to find that the explanations to these experiments will be a little easier, not quite as lengthy, and you'll get the information and it'll start to kind of make sense how you have to know what are the important ingredients, why are they in those tubes, like what makes those ingredients important, what's the pH indicator, if there is one, because most of these have a pH indicator, and then what does that color mean? What does that color mean? And that, usually can tell you, like, usually a certain color will equal positive. Yellow equals positive in these. Okay.
Are you, regarding the pH indicator, are you going to point to a neutral and uninoculated tube and ask what the pH indicator is, or how would
Yeah,
you-
I mean, I could ask that on the test too. I could say, what makes phenol red broths? What chemical makes a phenol red broth change colors? You could say, oh, it's the pH indicator called phenol red.
Okay.
Or I could just say, what is the pH indicator in phenol red broth? And that's even funnier when I ask someone, what's the pH indicator in phenol red broth?
Yeah.
And they don't know it's phenol red because they can't remember that phenol red is the name of the pH indicator, even though it's in the name of the test. Whereas the oxidation-fermentation test, if I said, what's the pH indicator? That's a little tougher. You've got to remember, I wrote bromophenol blue. Yeah, that's a little, I mean, some of them you have to write it down and just, memorize it so that you get that point on the test.
So none of this is going to be on the exam though, right, that we did today?
Oh, yeah.
Practical.
It will be?
on the practice,
Yeah.
but not the
Practice.
quiz.
Not
It won't be on the quiz. The quiz will be over just those 5 items.
Okay.
Okay, which I just didn't say just, that's a lot of, those are quite a few items that you're gonna have to go back and try to review. And then this stuff and, well, that stuff, I don't know, that's gonna be on the quiz. These items won't be on the quiz. We'll wrap those up on Monday.
This quiz?
This will be on the quiz coming up on Monday?
Yeah, phenol red broth glucose.
Oh, yeah.
Yeah,
So for the lecture exam, it's multiple choice and short answer, right?
yeah, exactly. Well, multiple choice, matching, true, false. Now, true, false can be brutal. One false word makes it false. Okay. But I usually make my true, false questions very false or very true. I mean, they shouldn't be super tricky, but there will be a few true, false questions just because it's a different style to test you on. And there'll be some matching, which matching are kind of the easiest. They just,
Process of
you'll have 5
elimination,
items,
yeah.
5 choices,
Yeah.
and you just need to match them.
Is there any like fill in the blank?
No, I'm going to do short answer. So, and those you even have ahead of time.
Yeah.
Like, I give you the 8 possible short answers, or 8 or 10.
10, yeah.
10, yeah. So I'll pick a couple. And if you do all of them though, that's good because that'll help you to answer some of the multiple choice questions and the matching and so forth. Yeah.
Is it Scantron or is it?
It's Scantron.
It's a Scantron. Okay, but what about with the short answers though and things like?
Those will be just written.
It'll be a combination of Scantron and.
There'll be
written
2
work?
pages on the back of the exam that are blank.
Okay.
They'll be just stapled to the exam.
Oh, I see. Okay.
And you'll
That'd
just
be the
turn the
questions.
page and there'll be those questions.
Okay.
Yeah.
All right.
Yeah.
I know.